about mimicking the sense of smell through artificial olfactory systems using proteins that are present in the real olfactory systems from many animals. Of course, those proteins all detect different odors. So this paper basically summarizes all the techniques that are used to develop those proteins and peptides, and of course the main obstacles that are present in that field.
So there is a trend in moving to combinatorial techniques in the discovery of new volatile binding peptides. These techniques allow researchers to test a very high number of molecules, increasing the success rate of finding new binding peptides.
Also, the progress in the biomolecule immobilization is increasing the reusability and the shelf life of the biosensors while still maintaining the desired selectivity, which is important. And this is also being achieved through the use of carbon nanotubes, gold surfaces, and other structures that mimic the cellular membrane.
So this study is very important because it summarizes the development of peptides and proteins for their application in gas biosensing.
Recently, we published a review on the technologies used in clinical microbiology to identify and obtain the antimicrobial susceptibility of pathogens bacteria cause of infective process. The methodologies currently used, however well established, show disadvantageous time and cost, being often too slow before achieve result. We review the well-established procedures, focusing on the other hand on the emerging and future technologies advances already available or still in process of optimization. These last represent powerful tools in our hands in order to decrease the healthcare costs associated with microbial infection and save life.
Bacteria and other microbes release volatile molecules to the environment as a result of their metabolic activities. Several research works have demonstrated that there are hundreds of these molecules released by bacteria, and sometimes the molecules are the same. So what we wanted to answer was if there is a set of volatile molecules that can work as a fingerprint and that can help us to identify microbial species. We developed a classification system, automatic, that uses artificial intelligence to discover from all the volatile molecules which are important to classify bacteria and other microbes. Our results are important to help to develop new diagnostics tools that can help diagnose infections. Currently, infections may be very difficult to treat because once a patient has the symptoms of an infection, doctors need to collect a biopsy, grow the biopsy in vitro, and then do several tests to discover what species of bacteria is causing the infection. And this may take between 1 day or 1 week. And in the meanwhile, besides taking a lot of non-specific antibiotics, the patient may get worse or even die. So imagine that we can have a device that simply sniffs out the biopsy and can identify the bacteria.
Albumin is a protein present in our blood. It is many times administered as a therapeutic agent for conditions such as hypovolemia, shock, surgical blood loss, and trauma. Due to its biopharmaceutic value, the aim of this work was to discover a new, less expensive and reliable synthetic ligand for albumin purification. Inspired by the ways albumin interacts in the body with other proteins and with other drugs, we used a rational approach and combinatorial methods to find a small synthetic molecule that we named A6A5. This new ligand was used in solid matrices for the affinity purification of albumin from human plasma. Given the position where it binds albumin, the new ligand can be explored as a spacer to bind drugs to albumin and in this way improve its circulation time in the blood as albumin drug conjugates.
So I won the poster prize on this poster right here, on the International Conference for Peptide Materials on Biomedicine and Nanotechnology, or PEPMAT for short. And this poster presents part of my PhD work on affinity materials, which are either gels
where the network is maintained by the presence of two molecules, the affinity pair, which bind together and form the hydrogel. It was important because it gave me the opportunity to present my PhD work, so I'm in my last year, and to see that at what point I was at that moment and how it could influence the scientific community to use it in their own research.